Hello students, welcome to the new lecture, and in this lecture we are going to discuss about the new topic that is vacuoles. And by the way, good afternoon everyone, good afternoon. And now we are going to discuss about the vacuoles. So in the class we have discussed about different types of the cell organelles. So let us see, this is a cell organelle. If you consider this is a cell organelles will have the nucleus. It has a endoplasmic reticulum, rough endoplasmic, smooth endoplasmic, and also it has a Golgi apparatus. Okay, mitochondria, ribosomes, etc. So these all are the cell organelles that you can see inside a cell. Okay, so we have completed chloroplast, we have completed endoplasmic reticulum. Okay, endoplasmic reticulum. We have completed the chloroplast. We have completed mitochondria, etc. So these all are the things that we have discussed already. And by the way, if you have any doubts or any questions, feel free to post in the live chat section, and we will discuss those doubts and we will clear those doubts and questions. Okay, so post your doubts or questions in the live chat section. So okay, fine. So we have completed these cell organelles. Now we are going to discuss about other cell organelle, that is vacuole. Okay, so what is meant by vacuole? Vacuole is a large and empty spaces in the plant cell. Okay, empty spaces in the plant cell, they are going to occupy entire cell, most of the cell. Okay, you might see the vacuole in the previous classes. In the previous class we have you might have observed. So this is a cell and this is the nucleus, and here will be the vacuole, large vacuole. Okay, if you have the textbook, open it and see. So this is the vacuole. So what is the function of this vacuole? So the function of this vacuole is to store water. Okay, first important thing is to store the water. Next, waste materials. It is also going to store the waste materials, and it is occupying the entire cell. It is going to occupy the entire cell. That is the main things that you can see in the cell. Okay, it is going to store water. It is going to store waste materials. It is going to store other substances also. It is also going to store other substances, and it is fluid filled organelle. It is filled with fluid and also some other components. Okay, and vacuoles are filled with fluid and also maintains turgor pressure. So what is turgor pressure? You might have seen the plant cells are tough. Okay, the plant cells are tough, and the plant cells are going to exert some pressure. It means, let us consider this is a cell. Okay, cell of a plant, cell membrane. Above the cell membrane there is a cell wall. Okay, so there is a cell wall. So what happening here over here? So here there is a vacuole. Let us consider this is a vacuole. Okay, this is the nucleus, and this vacuole, when it is filled with fluid like water, it is going to exert some pressure on the cell membrane. Okay, it is going to exert some amount of pressure on the cell membrane. So the pressure exerted on the cell membrane and the cell wall is known as turgor pressure. So remember, turgor pressure is known as stiffness of the plant cell due to the water, due to the presence of the water. That is known as turgor pressure. So, and sometimes it is going to export unwanted substances. What is meant by unwanted? Unwanted means that is not required by the plant. So plant don't require these components. So these components are known as unwanted substances. So these unwanted substances are removed from the plant. Okay. So not how the plant is going to remove unwanted substances? By leaf fall. Okay, so sometimes leaf will grow. When the leaf becomes old, the leaf cells will fall. Leaf will fall down. Okay, so when the leaf fall down, so what will happen? Already each and every leaf cell has the vacuole. Okay, already each and every cell has the vacuole. So when the leaf falls, what happens? The vacuole is also gone. Vacuoles has some amount of unwanted substances. So by this process, the plant cells are going to get rid of unwanted substances. Okay, so these are the materials. These are the process how the plants are going to eliminate waste materials, how the plant cells are going to eliminate waste materials, and how they are going to produce, how they are going to maintain turgor pressure, and so on. Okay, so this is about vacuole in a plant cells. And animal cells also going to have vacuoles, but very small. In animal cells, vacuoles are present, but very small, and some cells may not have the vacuoles. So this is about animal cells vacuole. Okay, there is not much important role in the animal cells about the vacuoles. Okay, So this is about it. And next is observation of vacuoles. So to observe vacuole, we need the aim of this experiment is to observe vacuole in the cactus plants. So what is a cactus? Cactus is nothing but it is a plant, desert plant. It is also xerophyte. Okay, you might have studied in the 8th class what is xerophyte. Xerophyte is nothing but these are the plants that live in the areas where there is less water. Okay, xerophyte. Okay, zero. The term zero means less water or no water. Phyte means plant. So it means the plants that are going to live in the areas of less water conditions, they are known as xerophytes. Okay, in this activity, in this experiment, we are going to study about the vacuoles of the cactus plant. So for that we need a cactus plant. Okay, we need water, we need watch glass, saffron in solution, microscope, scalpel. Okay, these are the things that we need for this experiment. Next, take the cactus plant. Okay, take a small stem and cut the stem into thin cross sections. Let us see, this is a stem. So this is stem of the cactus plant. So cut the cross section very, very thin. Okay, in this way. So this is small thin stem of the cactus plant. So now place this cactus stem, okay, in a watch glass containing saffron in solution. Okay, containing a saffron solution, and wait for some time. Then take out this saffron solution, place it on the slide. So this is the microscopic slide. So place the cactus layer. Okay, layer a small cross section of the cactus stem. Okay, so then add few drops of the water. Okay, so that to remove excess amount of the stain. Okay, then after it, keep the slide under the microscope and observe. Okay, so after observing, when you observe this under the microscope, you can see the cell. The cells are present in this way. Okay, the cells are present in this way, and in the cells you can see large sac-like structures. So large sac-like structures. These sac-like structures are known as vacuoles. So these vacuoles are very, very important for the maintaining of the turgor pressure, maintaining of the water or storing the water, storing waste materials, and also storing other unwanted substances that is in the vacuole. Okay, this is important organelle about the plants in terms of storing materials. Okay, so this is our observation, sac-like structures that can be seen in under the microscope. Next one will be the vacuole. So what is vacuole? Vacuoles are nothing but, just now we have discussed about it. Vacuoles are the organelles, the sac-like structure, they are observed under the microscope. Okay, this is our result. Okay. So next topic is are cells flat? Okay, are cells flat? If you observe the cells under the microscope, okay, this is a microscopic slide. Okay, and on the slide, if you put some cells, okay, and observe, if you put some cells and observe how they look. The cells looks in the 3 dimension. The cells look like in 2D. It means they appear like they are flat. Okay, they appear like they are flat on the slide. Okay, generally, actually, the cells are not flat. Cells are not flat. Cells have some dimensions. Cells have some properties. What are those properties? They have thickness. They are thick. They have some breadth. Okay, they have some length. And also they have some thickness. So these are the properties of the cells. Each and every cell has some length. Each and every cell has some length, breadth, thickness. Depending upon the type of cells, they may have different length, different breadth, and also different thickness. Okay. So this is about the cell's size and structure. And also remember, the cells also have some kind of structure. Okay. So thickness of the cells cannot be observed clearly under the microscope. Generally, you cannot observe the thickness of the cell, okay, and also its properties. But there is one small method so that you can understand the breadth of the cell, okay, breadth and thickness of the cell. So how do you observe? While observing under the microscope, slightly change the focus of the microscope on the tissues and try to view from the sides. Okay, try to view from the sides of the slide. Okay, then If you reduce such kind of intensity of the light, then the observation will be clear, and you can see the dimensions, or you can see the thickness of the cell up to some extent. Okay, so this is a small method to observe the cell thickness. Okay, so cell organelles are the structures organized in specific way and for the proper functioning in the cells. Already we have discussed the cells have some properties. What are the properties of the cells? So they have some size, thickness, they have some breadth, they have some shape, and etc. Along with this, they have the biological functions. Along with that, they have the biological functions. What are the biological functions? Each and every cell is going to have some function. Okay. So each and every cell is going to have some function. Each and every cell organelle is going to have some function. Example, take the mitochondria. Okay. Take the mitochondria. What the mitochondria is going to do? It is going to produce ATP. ATP means adenosine triphosphate. So this adenosine triphosphate is going to provide energy for the cell. Next, chloroplast. It is also cell organelle. It is also cell organelles. What is it is going to do? It is going to do photosynthesis. Okay, it is going to do photosynthesis. And nucleus. It is the commanding center of the cell, and so on. Okay. So these all are the different types of the cells, okay, and the different types of cell organelles, and they have some particular function. Okay, so based on it, we can understand that cells have some properties like size, shape, length, breadth, etc. They have some 3D shape, okay, that you can see in the diagram. So the first diagram here is the nerve cell. It is a nerve cell. It is a normal cell. Okay, normal cell of the plant. Okay, or animal. So you can see they have the three-dimensional structures. Okay, so these are the properties of the cells. Okay, and cells, with the help of these biological functions, cells will be in living condition. They work for better. Okay, so next is where do cells come from? Scientists have many doubts. Their doubts is, where do cells come from? Cells are going to come from the pre-existing cells. So the two scientists formulated means they did some experiment to identify where cells are coming from. So to explain where do cells come from, they proposed a theory. That theory is known as cell theory. Okay, that theory is known as cell theory. So what is cell theory is going to say? First of all, who proposed the cell theory? So cell theory was proposed by the two scientists, Matthias Jakob Schleiden. Okay, Matthias Jakob Schleiden, and also he is a botanist, and also another scientist is Theodor Schwann, a zoologist. So Matthias Jakob Schleiden, he belongs to the 1804 to 1881. Okay, in that region. Okay, so in his lifetime he proposed a cell theory. So Schwann was 1810 to 1882. So this is his lifetime. So when they are alive, so while they were doing research, they found some kind of cell theory to explain about the cells, how they are coming from. So they explained about cell theory, but there is small problem in their explanation. Their cell theory could not explain about the formation of new cells. They explained well, but their theory could not explain the formation of new cells. So this is one small drawback. So to overcome this problem, there is another scientist. Another scientist is Rudolf Karl Virchow. He belongs to 1885. In the 1885, he modified the cell theory of these two scientists. Like Matthias Schleiden and also Theodor Schwann. So he modified the theory of these two scientists, and he gave the final conclusion and final shape to the theory. Now the cell theory is perfect. And this is the cell theory. So their cell theory is modified by the Rudolf Karl Virchow, and the cell theory was first started by the Theodor Schwann and also here you can see another scientist, Matthias Jakob Schleiden and also Theodor Schwann. They proposed the cell theory, but so final shape was given by the Rudolf Karl Virchow in the 1885. Okay. So first, the cell theory, the two cardinal principles of the cell theory. What is cardinal? Cardinal means important. Okay, the cardinal means important principles and basic principles. So cardinal principles means important basic principles. What are the cardinal principles of the cell theory? First principle. The first principle is all organisms, all living organisms are composed of cells and products of the cells. Okay, so if you take a cell, okay, if it is a plant cell, what the plant cell is going to contain? The plant cell is going to contain some sugars. Okay, it is going to contain some sugars. It is going to contain proteins. It is going to contain vitamins, etc. It is also going to contain DNA, etc. So it means the cells is going to contain the products of the cells. The cell contains product of the cell. What are the products? Products like DNA, RNA, sugars, proteins, vitamins, etc. Okay, so cell contains all the products of the cells, all the products of the cells. So this is one principle. All living organisms are composed of cells and also products of the cells. If you take ant or if you take the human being, how the human being is made? Human being is made from the cells. You are containing the number of uncountable cells, and also each and every cell of your body is going to contain the product of the cells. Okay, that is one thing. That is first principle. Second principle is all cells arise from the pre-existing cells. Let us consider this is a cell. How the cell came? This cell came from the another cell, the parent of this cell. So how the cell came? This cell came from the another cell, and so on. It means the new cells are arising from the pre-existing cells. Okay, so whatever the cells that you have, how they came from the pre-existing cells. Okay, it means mother of the cells. Okay, so daughter cells, that this is the parental cell. Okay, this is the parental cell. Okay, the parental cell, when they divide, what they are going to divide, what they are going to form, they are going to form the daughter cells. These are parental cell and these are the daughter cells. Again they are going to divide. Okay, they are going to produce even more daughter cells. So by this way the cell theory is going to explain about the formation of new cells. Okay, and I hope this lecture is helpful to you, and if you have any doubts or questions or anything needed to explained, feel free to post in the live chat section and we will discuss about it. Okay. Okay, and by this theory, with the help of cell theory, the entire chapter is completed, and we are going to discuss next topic. Okay, that is second chapter. We are going to discuss second chapter. So in the next class, that is tomorrow. Tomorrow we will discuss the next chapter, and this is the notes. You can copy those points. Okay, it can be used as a notes also or class work. Okay. Okay, then we will discuss the next class tomorrow.